All right. Good morning, everyone. It is my pleasure to be here to introduce how to genotype CFTR, a gene of the cystic fibrosis with the next generation sequencing data using a method called T1K. Let's start with some key facts about cystic fibrosis. CF is a life-threatening genetic condition that primarily affects our lung and digestive system. And CF is caused by the mutations in cystic fibrosis transmembrane conductance regulator, or CFTR gene. And these mutations disrupt the chloride channel in our epithelial cell, which regulate the movement of water and salt across the cell membranes. So when mutations occurs, it disrupts this essential function, leading to thick mucus production and chronic inflammation in multiple organs in our bodies. In the United States, about 40,000 people are living with CF today, and about 1 in every 30 people is a carrier. The CFTR gene is located on chromosome 7, and to better understand and manage the disease, one key resource is called the CFTR2 project. This comprehensive database offers a curated list of CFTR gene alleles where every allele can contain one or multiple mutations. Among over 1,000 mutations identified, the F508 deletion variant, which involves a deletion of 3 nucleotides, is the most common CF disease-causing mutation, accounting for about 65% of the cases worldwide. Therefore, genotyping CFTR gene is very important for confirming the CF diagnosis and determining the best therapeutic approaches. However, a major challenge remains. There is no standardized method for accurately genotype the CFTR gene, especially on the sequencing data. We hypothesized that a genotyper for highly polymorphic gene like HLA and KIR gene can be extended to genotype CFTR. These immune gene families can have thousands of highly similar alleles and require specialized method for genotyping. The method that we are going to extend is called T1K, which is developed by our lab. And the idea of T1K is to find the most abundant alleles based on the data. Here, I want to show how flexible T1K is for both RNA sequencing data and the genomic data. For RNA-seq data, we build the reference sequence by just concatenating the exon sequences. And for the genomic data, we add the flanking intron sequences as the anchor. Instead of showing our own evaluation results, I present results from the unbiased third-party benchmark studies. HLA genotyping is important in immunology studies, so there are numerous benchmark studies comparing genotyping methods. There are 2 such studies after T1K's publication, and T1K ranked top in both of them. Motivated by T1K's proven accuracy and intuitive command line interface, we can now apply its read count abundance model and flexible reference designed to deliver precise and scalable CFTR genotyping. And the reference database is created from the CFTR2 database. To illustrate this process more clearly, here's a pipeline showing how we build the custom CFTR allele database for T1K. So we start with obtaining the CFTR reference sequence and retrieving a comprehensive variant catalog from the CFTR2 database. And each variant name is then parsed and normalized to prepare for the sequence induction. In this step, we determine the mutation type, dissect the notation, and translate those cDNA positions into genomic coordinates. In the next step, we take each normalized variant and induce it directly into the wild-type CFTR sequence to create a full-length mutant allele. For a simple deletion, we excise those bases, and for a simple substitution, like G to T, we replace the reference G with T at the exact genomic coordinate. While CFTR2 catalogs complex alleles, which means that one allele can have multiple variants, it is far from complete. Therefore, we generated complex allele sequences by combining two individual alleles on the same reference backbone. For example, F508 deletion plus a second point mutation here to model the real patient genotypes. Then we translate every induced CFTR allele into its amino acid sequence. By comparing these protein sequences, we can group the alleles with identical or functionally equivalent changes into unified protein families. So it will make the downstream interpretation more intuitive. Each allele is also assigned with a unique allele ID, which helps to track and reference the specific variant. However, users also have the flexibility to select the amino acid family ID for broader categorization based on the functional similarities. Once annotated, we then bundle these sequences and feed it into T1K. So running T1K against this custom reference then yields a straightforward output. For each sample, we report two most abundant alleles with their abundance estimate and the quality scores that reflects the genotyping confidence. Here, we have the CFTR0001 as the amino acid sequence for germline, and all CFTR0002 alleles contain F508 deletion mutation. We next test T1K's performance on real datasets. We genotyped CFTR using T1K in bulk RNA sequencing data from 21 CF patients, including their bronchial and nasal samples. We also randomly picked 30 individuals from the 1,000 Genome Project for their whole exome sequencing data to represent a general non-diseased cohort. T1K generated confident allele calls in 61 out of 71 samples. 59 were called F508 deletion only, 1 heterozygous, and 1 non-F508 deletion. 10 samples just had insufficient read coverage. These results illustrate that T1K can robustly recover clinically important CFTR genotypes directly from the RNA sequencing data. And none of the controls has F508 deletion genotyped. Here, we provide a visual confirmation of the F508 deletion cause by inspecting the RNA sequencing data, alignments, and the IGV. As we can see, the F508 deletion mutation is indeed present in these patient samples. And the new finding that we are excited to share is that we observed a paired mutation within the same CFTR allele in these samples. Specifically, in addition to the F508 deletion mutation, we observed a point mutation in an earlier position. These findings underscore the complexity of the CF genotyping and also supports why we combine the variants into the more complex allele set. I want to highlight that this complex allele is a novel finding from T1K, and it is not cataloged in the CFTR2 database yet. This complex allele captured by T1K is also supported in the literature. For control, we looked for germline unmutated sequences, and T1K did not find any patients expressing germline sequences. On the other hand, all but one controls have germline sequences. Here is a visualization confirmation for general population, and in both samples, the realignment show uninterrupted coverage across the F508 deletion region. To wrap up, T1K delivers highly accurate CFTR genotyping in both simple and complex pair mutations. And our custom reference pipeline automates everything from the variant name normalization to the allele induction, making the reference library creation both reproducible and user-friendly. Also, T1K is flexible, and it proves that it works equally well on the RNA sequencing data and the exome data. In short, T1K offers a great solution for genotyping CFTR. And looking ahead, we plan to study CFTR alleles in CF by profiling transcriptomic differences across different CFTR alleles. And also integrating T1K's results with patients' clinical records and metadata will let us correlate specific mutation profiles with drug responses and disease trajectory. This integration lays the groundwork for truly personalized precision medicine strategies in the cystic fibrosis care. We also plan to extend T1K beyond CFTR to other polymorphic genes, building the custom allele references for genes like BRCA1 and CYP2D6, which is a pharmacogene. Finally, I would like to thank to my advisor, Dr. Lee Song, for the invaluable guidance and support throughout this work. And I would also like to thank the faculty members at Dartmouth. And finally, I deeply appreciate the Cystic Fibrosis Foundation for their funding, which has made this research possible. Thank you so much for your attention, and I'm happy to take any questions.
Any questions here in the audience or any questions from online?
Thank you. Great talk. What would it take, though, the last point, generalizability of your pipeline? How much effort would that be to, or can you have like something like a more general pipeline where, you know, people say, okay, this is my polymorphic gene. I'm interested in doing predictions. Can I use your framework to do that for any polymorphic genes of interest?
So this is a great question. For the CFTR gene specifically, what we can pull up from the CFTR2 database online is the variant name, which include the precise cDNA positions and also some mutation types that's already included in the variant name. So that's how we splice that notation out, and then to identify like what type of mutation it is, and also like what position that mutation is. So if, for example, you have other genes that have the similar variant name structure, we can definitely test on those with this pipeline. And also for other type of variant names that you may have, we may need a little bit time to look into it. So I think it's also pretty promising that it can be generalized to other polymorphic genes.
Thank you.
Thank you.
Very nice presentation, and also very nice tool. What does T1K represent or mean?
Oh, that's a great question. So it has its own publication name, but I'm happy to share a little bit story about how my advisor who developed T1K is about the cells and what? One, is it the transformer, right?
The T1K terminator.
Oh, yes, the terminator.
All right, great.
So I like this interpretation better.
So is it available as a package for people to download and run as a command line app, or?
Yes, so basically all you need to do is go to the GitHub and follow the instruction there. It's super easy. And also like you basically just need to run the command line on it, and also like super straightforward.
One last question. Does it cover any structural variants?
Oh, sorry, what is that?
Structural variants.
Structural
Variant.
variant.
Yeah.
Okay, that's a great question. So we do have over 9,000 variants that's built in the reference library, but also like after grouping them by amino acid sequence, we ended up with 6,000 alleles or structural variants. So you can also like set different threshold for combining those individual alleles, and will result in different numbers of the reference data or library.
Thank you very much.
Yeah, thank you.